Today we're going to talk about mutations and how they can generate genetic variation. The goal is to define a mutation, identify the major types of mutations, and explain the effects a mutation can have on the survival of an organism. So first, what is a mutation? Mutations are changes to the DNA sequence, and they occur through errors that are made during DNA replication. Changes to the DNA modify the existing genes on the DNA. Remember, a gene is just a segment of the DNA. So a change to the DNA would change the gene, and this can create variation in the form of a new allele, a new version of that gene. Now, what are the types of mutations that can happen? So broadly speaking, there's going to be two types that we'll discuss. The first is called a point mutation. A point mutation is a change in just a single nucleotide. And there are going to be three kinds of point mutations. There's an insertion, substitution, or deletion. So an insertion just adds a base. So for example, say I had this sequence of letters in the DNA, ATG. If an insertion happens, a new base is inserted or put in there. So ATG could become AT, and then you added a C right here, G. So you just inserted a letter. In this case, it was the letter C. A substitution replaces a base. So go back to the original sequence I had, ATG. If a substitution happens, then you would switch out the T for some other letter. So I have now ACG. So this is where the substitution happened. And then a deletion removes a base. So if I had the letters ATG and I remove the letter T, now it becomes just AG. So those are the three types of point mutations that can happen. So point mutations, that's just one type of mutation. The other type of mutation is called a block mutation. And a block mutation is a change to a whole segment of a chromosome or a segment of the DNA. So there's going to be four kinds of block mutations: duplications, deletions, inversions, and translocations. I'm going to illustrate each one here. So a duplication is where part of a chromosome is copied. So here on the left, this represents a chromosome. Notice I have sections or segments of that chromosome labeled A, B, C, D, E, F. One of the segments just gets duplicated, it gets copied. So now on the right, you see the chromosome after the mutation has happened has an extra segment here. It's a copy of B. What about a deletion? A deletion is where a part of a chromosome is removed. So here's that original chromosome I have that I started with on the left. Notice I have the segment B, but after a deletion, that whole segment is removed. So now the entire section of that chromosome is gone. What about an inversion? So an inversion is where part of a chromosome is flipped around. It's kind of like spun around or inverted. So look at the original chromosome I started with, and you'll notice that segment B, segment C, segment D, and segment E, they kind of just imagine how hands on a clock can turn. It's just been turned around. Now E and D are on the top segment of the chromosome, and C and B are on the bottom part of the chromosome. So that would be an example of an inversion. A translocation, this is where segments of two chromosomes get exchanged with each other. So on the left here, I have two chromosomes now. And you'll notice that segment D and E here, notice it's capital letters, so capital D, capital E, and then lowercase d, lowercase e. These have swapped places now. Or at least the capital E and the lowercase e have swapped places with each other. That would be an example of a translocation. Okay, so I identified some types of mutations. Now let me talk about the effects of mutations. A mutation may change the way that a protein folds. So, if you'll recall the process of protein synthesis, that's where you use DNA to make RNA and you use the RNA to make protein. So, you go from DNA to RNA to protein. Well, if you change the DNA, some mutation there, then that's going to end up affecting the protein. And it could affect the way that the protein folds. And if it does change the way that the protein folds, that can affect and prevent that protein from carrying out its normal function. Now, if that changes its normal function, then there can be three possible effects on an organism. So, what are these effects and mutations on an organism? The effect could be beneficial. So, a beneficial mutation is a mutation that affects how the organism that actually helps an organism survive. If it's beneficial, then it's going to help the organism survive. Another effect is it could be neutral. So a neutral mutation is a mutation that doesn't help and it doesn't hurt the organism's chance of survival. This usually happens when the mutation doesn't change the sequence of amino acids in the final protein. And then the last type of effect that the mutation can have is a harmful one. A harmful mutation is a mutation that doesn't help an organism survive. In fact, it may make it less likely to survive. Now, most mutations end up being harmful, but occasionally they are beneficial. Let me give you an example of a harmful mutation. Albinism. So you might have heard of people that are albino. Albinism is caused by a mutation, and this is an example of an albino alligator. And this is a harmful mutation because when the individual is albino, they don't produce a pigment in their skin that protects them from the UV light from the sun. So this alligators actually can be easily burned by the sun and end up dying as a result. So this would be an example of a harmful mutation. So to sum up, we've defined what a mutation is. We've looked at the different types of mutations, and we've looked at the different effects that mutations can have on an organism.